Hello students, welcome back to the channel. In this video, we are going to study about the stem cells. We will be going to study what is the difference between multipotent, pluripotent and totipotent cells are there and how we are going to use the stem cells for curing the diseases. So let us start the video. See, first of all, what is the definition of stem cells you will give? You will say they are unspecialized cells. They can reproduce themselves indefinitely, and under appropriate condition they can differentiate into specialized cells also, either one type or more than one type. So this is the definition of the stem cell. If you see, this is one diagram. This is stem cell which is dividing into two. So it is retaining one cell is behaving as a stem cell, another cell, okay, the another cell is known as precursor cell. Either it can differentiate into fat cells, or it can differentiate into bone cells or white blood cells. It means they are the one which can replenish their own population, and they can make the specific differentiated pathway also. Because of this reason only, because of these two reasons, they can divide again and again to produce their own copies, and they are not fully differentiated. They can be differentiated into many types. That is why they are the most active area for research in biology and medicines. Now see, we'll start for the human. First of all, understand every living organ is sexually reproducing. They start their life from the one cell zygote, which is going to divide into 2 cell, 4 cell. And these cells, they are the one which are at this early stage of embryonic development. We will say that they are the one which are capable of dividing many times to produce large amount of tissues. They are very versatile. They can differentiate in different pathway also. So we call zygote as totipotent. Why we say totipotent? Because it can give rise to all type of cell in the body, including which make extra embryonic membrane or which make placenta. So zygote is considered as a totipotent, or the cells which are at up before blastular stage. Okay, after 4th day of the development in human, what we find, the cells are differentiated to make blastula. They are making the layer. If you know blastula structure, this is the trophoblast, which is going to give rise to extra embryonic membrane. They are the helping in formation of placenta. Okay, and this is the inner cell mass. This one which I'm using with the red color, these inner cell mass. So embryo develop from the inner cell mass. And the one which blue color which I have drawn it, that is your totipotent. So just let me highlight it. This is your inner cell mass. Clear, beta? And this is what? This is your trophoblast cells, which are going to make the extra embryonic membrane, or you can say they are going to make the placenta. Inner cell mass is the one which is going to give rise to the embryo. If you take out the cell from this stage, then that cell is known as pluripotent cells. They are also known as embryonic stem cells, because they can develop into any of the body cell types. Even they can differentiate as a germ cells also, but they cannot make the tissues which are surrounding the embryo. Clear, beta? They cannot make the tissues which surrounds the embryo. So you know, I told you outer layer is trophoblast, which give rise to the tissue which attaches the embryo to the mother, and inner cell mass give rise to the embryo. The cell of inner cell mass are known as pluripotent or embryonic stem cell. After this, when the complete development has occurred, then what happened? Slowly, what happened? Differentiation is going on, and they are becoming into the organism has become into the adult. So small number of cells still remain as stem cells in the adult body. They are present in the adult body, and they are very small number, and they are known as adult stem cells, also known as multipotent. Why we are saying they are multipotent? Because they can give rise to one specific cell type. Suppose you are finding the cells in the bone marrow. They can make all the cells of the blood, but they cannot make the muscle cell, or they cannot make the nerve cell. So these are multipotent, which can differentiate. Okay, now in the adult body, you are finding these adult stem cells in the bone marrow, in the skin and liver. and they are one responsible for regeneration and repair also. Please focus bone marrow, skin and liver, they are giving the power for regeneration and repair. But the one which is present in the brain, kidney and heart, they are only showing limited repair. Only limited repair they are able to show. The stem cells which are present in the adult body, they are very few in number and they can help. Few can help in both regeneration and repair, and few can help only in repair. So we are doing the research work in these types of stem cells with the embryonic or adult stem cells. Okay, what is the main aim? Main aim is to produce regenerated tissues. For example, the people who are suffering burn, we can regenerate the skin cells. Okay, we can do the regeneration because here your stem cells are present. They can be helped in the healing the diseases. The people who are suffering from type 1 diabetes, the problem is one in which the insulin deficiency is there, that we can cure it. Brain cells we can use to cure the disease, cure like Parkinson disease or Huntington disease. Adult stem cells, if taken from bone marrow, they can be source for the immune system cells in the patient whose immune system is non-functional. Okay, so development of adult cells, but problem is it is limited. Embryonic stem cells are having more versatile as compared to the adult stem cells. Because for most medical applications, adult cells are pluripotent. Sorry, embryonic cells which are pluripotent, they are capable of differentiating into many different cell types. So we are preferring the embryonic stem cells over the adult stem cells. But main thing is this, how to obtain the embryonic stem cells is to harvest them from the embryo, which is again raising the ethical issues or some political issues are there. So that is the only problem we are facing. Though they are having more potential, we know embryonic stem cells are having more potential as compared to the adult stem cells. Okay, the development potential of adult stem cell is limited. Embryonic stem cell is giving more future advantage. We can do a lot of research or we can cure many diseases with the help of embryonic stem cell as compared to the adult stem cells. Embryonic stem cells can be useful in the future to grow whole replacement organ also we can do it. And these types are used for therapeutics because we are using them as therapies for disease or other health related problem. So when our aim is to use these stem cells, cloning the stem cells, we are doing it to treat the disease, then you see it is therapeutic. Once again, see the difference in the diagram. Check it, you will find it. This is your embryonic stem cells. You can see better. This is the inner cell mass. If you are using the inner cell mass in the culture, you can differentiate either into liver cell or nerve cell or blood cell, anything you can differentiate. But if suppose you are isolating the adult stem cells, for example, bone marrow, they can generate only limited number of cell type. If you're taken from bone marrow, they can make only the blood cell. You can see this line only, they can make it. So what is the difference between them? If you see, embryonic stem cells, they show unlimited growth potential. They can differentiate into any types, but they cannot make extra embryonic membrane. They are more risk of becoming tumor cells than as compared to the adult stem cells. Less chances of genetic damage due to accumulation of mutations. They are genetically different. Likely, chances are this, they can be genetically different from the adult patient who is receiving the tissues. And main thing is this, if you remove the cells from the embryo, it will kill it unless only 1 or 2 cells are taken. That is, a few drawbacks are also there. But if you see with the adult stem cells, first of all, it is difficult to obtain because they are very less in number and they are very deep into the tissue. They are having less growth potential as compared to the embryonic stem cells. Less chances of tumor formation as compared to the embryonic stem cells. They have limited capacity to differentiate because if you are isolating from that particular area from bone marrow, they can make only blood cell. They cannot differentiate much. But they are fully compatible with the adult tissue because they have obtained from the adult stem cells. So rejection problem is not there. But main thing is this removal of the stem cells. If you see, that is a problem because they are deeply embedded, buried in the tissue. In case of embryonic stem cells, we studied if you remove, it will kill the embryo. But here, removal of stem cells does not kill the adult. So I hope so in this video, we try to focus what is stem cells, how are they producing their own copies, as well as they are producing the precursor for the cells so that they can differentiate into different types. Zygote, which can make every cell, including the extraembryonic membrane. We say zygote is totipotent. Blastula stage, in that inner cell mass can differentiate into any types but not extraembryonic. Then we say that is known as pluripotent, and which are present in the adult body. Only few numbers and few places that are known as adult stem cells or multipotent. I hope so this video will be helpful for you. Thanks for watching.